Hello everyone, welcome to the second part of chapter Neoplasia. So in the last class, we have discussed about the difference between benign and malignant tumors. So benign tumors are small, they are well demarcated, they are slow growing, non-invasive, non-metastatic, well differentiated. Whereas malignant are locally invasive, metastatic, poorly differentiated, large, poorly demarcated, rapid growing with hemorrhage and necrosis. Benign tumors are circumscribed, they have a capsule. Malignant tumors are poorly circumscribed, so they invade the surrounding tissue. Remember, metastasis is the first hallmark feature which helps to differentiate between benign and malignant tumors. Coming to epidemiology of the cancer, there has been some update. There has been some update in the epidemiology of cancer. Okay, so I'm going to use that updates only in the 10th edition. 10th edition updates. Fine? Yes. Okay. So a very important point that you have to remember here, as per the 10th edition update, the most common malignancy in India in males are lip and oral cavity cancer. Okay, and the second is lung cancer. In female, breast cancer first, followed by cervix cancer. So I hope you are clear with this. Please just remember those two. These are the two important updates. Fine? Yes. Okay. Let us go for the worldwide data. Okay, the worldwide data. As far as worldwide data are concerned, you can see the incidence of lung cancer is the most, followed by the incidence of prostate cancer is high in males, followed by lung cancer. Number one prostate, number two lungs. As far as females are concerned, still breast cancer stood number one. The most common cause of cancer death, it is because of lung cancer, followed by prostate cancer. And as far as female is concerned, again we have lung cancer only. Fine? Yes. Then another important update, hepatitis C virus has been begin to develop the hepatocellular carcinoma in the past 40 years, decades. Fine? Yes. Then, and remember, the decreased use of tobacco product is responsible for reduction in lung cancer death. Okay. And coming into the what are the environmental factors, we have infectious agents called human papilloma virus that is responsible for causing cervical cancer and head and neck cancer. I am just focusing the key points. Okay. Yes. Then coming into smoking, as far as smoking is concerned, it is one of the most important environmental factor which contributes to premature death in United States. So this is the geographical distribution. Okay, worldwide variation of cancer incidence in males. Okay, and as you can see here in India, as far as India is concerned, we have the lung and bronchus, but remember bronchus, but now it is lip and oral cavity cancer. Lip and oral cavity cancer is the predominant cause. Okay, predominant cancer, followed by lung cancer. This is the update. This is the 10th edition updates. Fine, I hope you are clear. Fine. Once this is done, see, this is the incidence of breast cancer. See, in India we have around 17.2 to 25.8. Fine. Then coming into what are the occupational cancers. Okay, please focus this table. Arsenic, lung cancer and skin cancer. Asbestos, mainly mesothelioma. What is the most common type of lung cancer caused by asbestos? It is the adenocarcinoma of the lung. Whereas the most specific cancer which is caused by asbestos is the mesothelioma. Benzene causes AML, acute myeloid leukemia. Beryllium causes lung cancer. Cadmium causes prostate. Chromium, beryllium, nickel, radon, all those causes lung cancer. Vinyl chloride causes hepatic angiosarcoma. This is vinyl chloride causes hepatic angiosarcoma. This is a potential MCQ question. Fine. So the common neoplasm of the infancy and childhood, we call it as the small round blue cell tumors. What are the tumors? Okay, which comes under the small round blue cell tumors. You have to remember this name, of course. Number 1 is Wilms tumor. Number 2 is neuroblastoma. Number 3 is retinoblastoma. Number 4 acute leukemia. Number 5 rhabdomyosarcoma. Fine. Yes. Then, what are the predisposing conditions for cancer? Some of the chronic inflammatory conditions are associated to have increased risk of causing cancer. Okay, number one is asbestosis and silicosis. I have already told you it is going to cause mesothelioma and adenocarcinoma of the lungs because asbestos fibers. We will see more detail in lung pathology. Inflammatory bowel disease has been in a long run. Okay, that too especially ulcerative colitis. In inflammatory bowel disease, we have Crohn's disease and ulcerative colitis. Out of that, ulcerative colitis is having very high malignant potential. It is known to cause colorectal carcinoma. Then we have something called as lichen sclerosis, which causes the vulva, okay, vulval cancer. Pancreatitis, pancreatic carcinoma. You know, one of the most common cause of pancreatitis is alcoholism, followed by germline mutations. Then we have chronic cholecystitis, which causes gallbladder cancer. Barrett's esophagus is one of the predisposing factor, and it is one of the it predisposes to esophageal carcinoma, adenocarcinoma. Sjogren's syndrome, malt lymphoma, because in Sjogren's syndrome, as I have already told you, there will be lymphocyte deposit, proliferation. Then Opisthorchis cholangitis causes cholangiocarcinoma. Then gastric ulcers causes gastric adenocarcinoma, hepatitis, hepatocellular carcinoma. Okay. Then chronic cervicitis, cervical carcinoma, and chronic cystitis, bladder carcinoma. Okay. Hepatitis caused by hepatitis B and C. Gastritis ulcer caused by Helicobacter pylori. Osteomyelitis, bacterial infection. Then next one is what we call it as chronic cervicitis. Cervical cancer is caused by human papillomavirus. I have already told you. And schistosoma hematobium causes bladder cancer. Fine. Yes. What are these precursor lesions means? Because they are associated with high risk of cancer. So chronic inflammation is one of the predisposing factor for malignancy. Fine. Okay. Then another most important thing, yes, this is what I have told you. Fine. And in endometrial hyperplasia, it is also one of the, because of the estrogen, okay, sustained estrogen stimulation causes what? Endometrial proliferation, and that is one of the endometrial hyperplasia is one of the most common precursor for endometrial carcinoma. We have type 1 and type 2 that we will see later. Fine. Yes. And another one is what we call as leukoplakia. Leukoplakia is nothing else but thickening. Okay, leukoplakia is nothing else but what we call as, it is the thickening of the squamous epithelium. Okay, it may give rise to squamous cancer. Okay, yes. Then villous adenoma, it may even give rise to colorectal cancer. Fine. Yes. Then coming into genetic predisposition and interaction between the environmental and inherited factors. Out of that, one of the most important genetic predisposition that I would like to mention here is the breast cancer. See, breast cancer is more in females who have BRCA1, BRCA2 mutations. I hope you all know about the actress Angelina Jolie. She actually has a familial inheritance of BRCA1, BRCA2 gene mutation. BRCA1 mutation that is responsible for her to develop breast cancer. Fine. Yes. So very high risk factor. Okay. Then coming into what is the molecular basis of cancer. See, actually, what is tumor? See, when a single cell undergo clonal expansion because of some genetic damage, what will happen? That these genetic damages are inherited, so it will pass to the daughter cell, and the same problem. That is why in families the cancer runs. Fine. Yes. Okay. So we have 4 classes of regulatory genes. Number 1 is called as growth promoting or proto-oncogenes. Next one is called as growth inhibiting tumor suppressor gene, and third one is apoptosis regulating gene, and finally one is the gene involving the DNA repair. Please remember this 4 set of gene: growth promoting proto-oncogene, growth inhibiting tumor suppressor gene. Okay, so when the growth promoting proto-oncogenes are overactivated, or the growth inhibiting tumor suppressor genes are inhibited, or there is an evasion from the apoptosis, or whether there is a problem in the genes involved in DNA repair, this all will finally leads to cancer. Fine. Yes. Okay. Tumor suppressor gene. Okay, I told you, yes. DNA repair genes. Finally, genomic instability. So we have something what we call it as hallmarks of cancers. Okay, what are the, see, this is how some of the mutations is responsible for cancer. See, there will be a normal cell which will be exposed to some carcinogens. Carcinogens are substances that will promote proto-oncogenes activation and inhibit tumor suppressor gene. So it will produce some genomic instability. Okay, and this mutation will be further undergo accumulation. That is what we call as driver mutation, and the cancer cell will further develops to the heterogeneous cancer. Fine. Yes. Okay. Then fine. So finally, okay. Next, so tumor progression. This is what we call it as tumor progression. Fine. Once this is done, See, I told you that DNA repair. See, children, in addition to this mutation, epigenetics, I have told you in the chapter genetics itself, epigenetics is one of the most important reason to contribute or which is responsible for the malignant properties of the cancer cells. We have two important epigenetic alteration. Epigenetics means there will be some chemical modification in the DNA sequence without some alterations in the there will be chemical modification of the DNA without any alteration in the DNA sequence. The most important thing is DNA methylation and histone modification. Okay, just to remember these 2 points. DNA methylation, it is one of the most important thing for responsible for colonic cancer development. Okay, yes. Finally, coming into cellular and molecular hallmarks of cancer, there are some 8 fundamental changes in the cell physiology which we considered as the hallmarks of cancer. Number 1 is if you know this, because molecular hallmarks of cancer is a straight away standard essay question most frequently asked in your prof exam. Self-sufficiency in growth signals, that means the growth signals will be continuously being activated, or there will be insensitivity to growth inhibitory signals, or whenever there is altered cellular metabolism, that means the tumor cells will undergo a metabolic switch. They will undergo aerobic glycolysis. Normally glycolysis does not need oxygen, whereas in tumor cells it will use what we call as aerobic glycolysis, and this phenomenon is called as Warburg effect, which we will see study in detail. Fine. And then evasion of apoptosis, then limitless replicative potential, sustained angiogenesis, because only whenever there is an increased vascularity, that is why most of the malignant conditions, all these are highly vascular tumors, am I right? They have very rich vascularity. Fine. Yes. So, and ability to invade and metastasis, and finally ability to invade the host immune response. All these are the 8 fundamental hallmarks of cancer. All these are the 8 fundamental hallmarks of cancer. I told you 2 important things. One is what we call as oncogenes. What is oncogenes? That means the genes which promote autonomous cell growth is what we call as oncogenes, and their cellular counterparts are called as proto-oncogenes. So normally the cell has to grow. Okay, even I, even you, all of us has normal oncogenes, but within the cell, okay, that is there are something which is similar to oncogenes, that is what we call proto-oncogenes. Normally all our proto-oncogenes are inactivated. Whenever there is carcinogens, these carcinogens will go and stimulate the proto-oncogenes. Fine. Yes. Remember, these oncogenes and all these are made up of some proteins which it will produce. They will encode some proteins after translation. They are called as oncoproteins. Okay, yes. So these oncoproteins are responsible for maintaining the growth signaling pathways within the cell. Okay, I hope these things are very clear for you. Yes. Okay, see. Next, this is a very, very important table. Okay, some of the selected oncogenes, their mode of activation. You should know this table because they will give you, okay, what is this growth, this oncogenes and tumor suppressor genes. You should know how to differentiate. P53, it is not an oncogene, it is a tumor suppressor gene. So you should know at least the most common, okay, commonest proto-oncogene, so that in the MCQ they will ask you which of the following or all of the following are oncogenes except. So number 1 is PDGF, platelet derived growth factor beta. Okay, that is mainly in astrocytoma. Fibroblast growth factor in stomach, bladder, breast cancer and melanoma. Remember, fibroblast growth factor, stomach, bladder, breast cancer and melanoma, all these are associated with what we call it as desmoplasia. I told you what is desmoplasia already in the first lecture. Then TGF alpha and hepatocyte growth factor, TGF alpha in astrocytoma, hepatocyte growth factor, hepatocytes, so hepatocellular carcinoma and thyroid cancer. Then we have the growth receptor family, that is ERBB1 and 2. ERBB2 is called HER2/neu. HER2/neu amplification occurs in breast cancer, whereas ERBB1 undergo mutation. This is amplification, this is mutation. This is one of the frequently asked MCQ question. In breast cancer, the ERBB2 does not undergo mutation, it undergo amplification. In adenocarcinoma of the lung, ERBB1 gene undergo what mutation. Then we have something called as FMS like tyrosine kinase 3, okay, that is in leukemia. Then RET in tyrosine kinase, that is most commonly in medullary carcinoma of thyroid and multiple endocrine neoplasia. Then ALK is in adenocarcinoma of lungs and in neuroblastoma. Then ABL, okay, that is already ABL translocation, okay, BCR ABL translocation occurs 922 Philadelphia chromosome, chronic myeloid leukemia, and at the same time when this undergo mutation, when it undergo translation, it will produce a CML. When this gene undergo mutation, it produces acute lymphoblastic leukemia. That is why BCR ABL fusion gene, it is not only produced in found in CML, it is also found in ALL. Fine. Yes. Then RAS is mainly for melanomas. Okay, RAS is mainly for, and Then NMIC, CMIC for, so CMIC, whenever you heard the word CMIC, you should remember that is Burkitt's lymphoma. NMIC, N for N, so neuroblastoma. Okay, cyclin dependent kinase, CDK4, very very important, glioblastoma, melanoma and sarcoma. Okay, yes. So now let us see about these proto-oncogenes, oncogenes and oncoproteins. So this is the pathway. So whenever there is a, you can see here the growth factor and you have a growth factor receptor. So whenever this growth factor and growth factor receptor is, growth factor binds to the growth factor receptor which is present on the cell membrane, it will activate, it will convert inactive RAS into active RAS, and there will be subsequent hydrolysis of GTP to GDP, and further, okay, this GDP will inactivate the RAS, so this process will stop. Whenever there is a failure, okay, what will happen? This RAS will continuously get activated, which will cause as RAF and MAPK. MAPK is mitogen activated protein kinase, okay, or PA3K, AKT, mTOR. mTOR is called as mammalian target of rapamycin. Okay, so PA3K, AKT, mTOR activation, which will further causes increased protein synthesis, which will, all this will finally, okay, at last it is going to produce what we call the cell cycle progression, and finally this will causes cell growth. And when this pathway get continuously stimulated, overstimulated, excessive activation, finally the result is cancer. Okay, yes, that is abnormal proliferation or abnormal cell growth. So once again I am telling, growth receptor binds with so growth factor binds with growth factor receptor, okay, which will causes inactive RAS to active RAS. There will be hydrolysis of GTP and this GTP undergo hydrolysis to form GDP. This GDP will further inactivate the RAS. Okay, suppose what will happen if the RAS is activated? It will either activate RAF, okay, or it will activate PA3K. When it is activating RAF, it will causes MAPK activation, transcription activation, MIC activation, cyclins activation, cell cycle progression, increased protein synthesis. When PA3K is activated, AKT activation and mTOR activation, finally this also produced increased protein synthesis. At last, finally all this will produce cell growth. Fine. So growth factors, already I have told you what are the growth factors we have. Yes, PDGF, platelet derived growth factor. Yes, what is the condition? Very good, it is glioblastoma. And remember, transforming growth factor alpha, okay, all this is in sarcomas, very very important. Yes, okay. And next is growth factor receptors. We have ERBB1. ERBB1, it is mainly for lung cancer. ERBB2, HER2/neu, breast cancer amplification. Fine, yes. Then of course we have another what is ALK receptors in adenocarcinoma of the lung and neuroblastoma. Fine, yes, okay, fine. Okay, so once this is done, then we have RAS mutations. We have HRAS, KRAS and NRAS. So RAS mutations, they are mainly in pancreatic adenocarcinoma, cholangiocarcinoma. Okay, I'm just highlighting that: pancreatic, cholangiocarcinoma, colon, endometrial, thyroid carcinoma. Okay, and okay, so already I have told you what is the pathway of RAS. Okay, that is the two pathway, no, RAF as well as P10. Okay, yes, sorry, RAS as well as in case of PA3K. Okay, fine. So GTPase activity, everything I have told you. And remember, RAS protein is also involved in what we call as familial neurofibromatosis type 1. Okay, yes, neurofibromin, it is an example of a tumor suppressor gene. Okay, that acts through negative regulation. When we come to tumor suppressor, I will tell you neurofibromin, it is a tumor suppressor gene. That is why I told you, you should mug up this table. You should mug up this table because they will give you a question: which of the following are proto-oncogenes? Which of the following are tumor suppressor gene? At that time, you should not feel confused. At that time, you should not feel confused. Fine, neurofibromin, it is a tumor suppressor gene, act through RAS pathway. Fine, yes. Then we have BRAF. See, BRAF, it is actually a member of the RAF family. Mutations in BRAF has been detected in hairy cell leukemia, melanoma, okay, colon cancer and dendritic cell tumors. Very very important. Every year in your PG entrance exam, they will give you which of the following cancer BRAF mutations are present. Number 1, hairy cell leukemia. Number 2, melanoma. Number 3, dendritic cell tumors, and number 4 are colon cancer. Okay, they also act through MAPK signaling pathway. Okay, then I had already told you about the PA3K pathway also. Fine, yes. Then in non-receptor tyrosine kinase, we have something what we call as ABL, BCR-ABL fusion gene. I told you 922 translocation in chronic myeloid leukemia, that is translocation. At the same time, when this ABL gene undergo mutation, that is responsible for acute lymphoblastic leukemia. Okay, yes. So we have lot of what receptor tyrosine kinase inhibitor drug like what olapartinib, okay, imatinib. All these has shown severe. Okay, I will write the spelling imatinib. Okay, these are tyrosine kinase inhibitors. So this has been shown to have very good efficacy against the treatment of chronic myeloid leukemia. See, chronic myeloid leukemia, you can see 922 translocation. Okay, remember the ABL gene. Even though we are telling BCR-ABL, the ABL gene is located on chromosome 9, and BCR is a breakpoint cluster. Okay, breakpoint cluster region, that is BCR full form. So this is 922. So whenever this undergo translocation, we call it as a 922 translocation. See, 922 translocation. Finally, this will causes what? Excessive activation of the tyrosine kinase. This is responsible for activation of growth factor signaling pathway. In Burkitt's lymphoma, we have 814 translocation. Okay, this is MYC gene, which is present on chromosome number 18, undergo translocation. It is translocated to chromosome number 14. Chromosome number 14 already have something what we call as IgG gene. Okay, IG stands for immunoglobulin gene. So this will causes increased MYC protein, increased expression of pro-growth genes. Don't worry, we will see this in hematology. Okay. And we have something what we call as JAK-STAT signaling pathway. Just remember one point. JAK-STAT signaling pathway has been known, or it is implicated in the pathogenesis of some of the important neoplasm. That is, number one is called as myeloproliferative neoplasm, especially polycythemia rubra vera or polycythemia vera, essential thrombocytosis, and primary myelofibrosis. So JAK-STAT signaling pathway, it is also a type of non-receptor tyrosine kinase, whereas ERB1, ERB2, HER2/neu, ALK receptor, all these are receptor tyrosine kinase. Whereas non-receptor tyrosine kinase, we have JAK-STAT pathway or JAK2 or JAK-STAT signaling pathway is implicated in the pathogenesis of myeloproliferative disorder, particularly polycythemia vera, essential thrombocytosis, and primary myelofibrosis. Fine, I hope things are very clear for you. Okay. Then MYC. See, the MYC proto-oncogene, it is expressed in almost all. Okay, MYC and JUN. I am writing here MYC and JUN, JUN. Okay, this is I have explained in first chapter. MYC and JUN are the two important transcription factors that are responsible for the regulation of the cell growth. Okay, this is present in all eukaryotic cells. Fine. Yes, this MYC, what it is going to do? It is going to activate the cyclin D. Okay, it is going to upregulate the gene expression. Because of all which what happens, there will be cell growth and continued cell growth. Okay, and the most important thing, remember, Burkitt's lymphoma, we have c-MYC upregulation or c-MYC amplification. N-MYC. Remember, N stands for N, that is N-MYC is present in neuroblastoma. N-MYC amplification occurs in neuroblastoma. L-MYC, L for lung. Okay, so that is L-MYC in small cell cancer of the lung. Okay, just remember this thing. Okay. Then we have a lot of cyclins and cyclin-dependent kinase. See, this is N-MYC. Okay, yes. Cyclin-dependent kinase. Out of this, just to remember some of the important features only. Okay, you already know this the role of cyclins in the acting as cell checkpoints. In the cell cycle, we have two important checkpoint children. One is G1S transition and the other one is G2M transition. Okay, yes. So whenever there is a gain of function mutation in the cyclin D genes and in cyclin D kinase 4, CDK means cyclin D kinase. Okay, CDK4. Okay, it will promote G1S progression. Normally at G1S the cell cycle should stop, but here because of gain of function mutation, the cell cycle is not stopping at G1S, it is progressing. And here this is the problem which occurs in melanoma. I am writing here. See, this is melanoma, sarcoma, and glioblastoma. Melanoma, sarcoma, and glioblastoma, MSG. Fine. Yes, and whenever there is a loss of function mutation, what will happen? It will inhibit the G1S progression. This is also seen in case of pancreatic carcinomas, okay, glioblastoma, acute lymphoblastic leukemia, bladder cancer, small cell cancers of the lung, all those things. Fine. Yes, so this finishes oncogenes, oncoproteins, and upregulated cell proliferation. Next is insensitivity to growth inhibition. Here comes the role of tumor suppressor genes. Okay, here comes the role of tumor suppressor genes. Fine. Okay, I will discuss the tumor suppressor gene in the next video. In the next video, we will be discussing tumor suppressor genes. Okay, fine.